Guys, welcome back. We're going to continue our unit on DNA. Today we're going to be talking about mitochondrial DNA and STRs. Alright, so let's look at mitochondrial DNA. We've learned a little bit about this so far, but we're going to look at a little bit more. Okay, so of course mitochondria are organelles that turn sugar into energy within the cell, and every mitochondria has their own DNA. The mitochondrial DNA is different from the DNA in our nucleus. It is unique. It is ring-shaped. A ring-shaped DNA is called plasmids. Mitochondria are sort of like prokaryotes in that fact. It has less protection from mutation because it is not bound in a nucleus. It only has 37 total genes, and it has only about 16,000 base pairs. It is haploid, meaning there's only one, and not diploid. Okay, so you only have one set. Okay, so there's only a little bit of mitochondria in the human sperm, and during fetal development, most of it gets destroyed. So your mitochondrial DNA, as we've talked about before, comes solely from your mother's egg. So a male's mitochondrial DNA comes from his mother, but he will not pass down his mitochondrial DNA to his offspring. So here we have a pedigree which shows the progression of mitochondrial DNA. At the top we have a mother who's circle and a father who's square. The mother has the mitochondrial DNA. She gives it to both her offspring. She has a girl and a boy. The girl will continue to pass it on to all of her offspring, and her daughter will pass it on to her offspring. But you notice that that passing down stops at the male. Alright, so moving on from mitochondrial DNA, that was quick. Alright, we need to start talking about microsatellites and short tandem repeats. So, alright, so the human DNA that forms genes that code proteins or RNA there's a very little of it. Over 98% of the human DNA is considered non-coding. It doesn't have, I wouldn't say it has a purpose. I think its purpose is unknown to us at this point. But about 2% of the human DNA we know actually codes for stuff. Okay, but because of our lack of understanding, for a long time the non-coding DNA got the nickname of junk DNA. But however, we're now coming to this understanding that it may have important functions that we just haven't discovered yet. So in non-coding DNA, there are sequences of about 2 to 6 base pairs, and these are called microsatellites. Okay, microsatellites become used in short tandem repeats. Okay, what does short tandem repeats mean? Okay, so like we said, they're usually 2 to 5 base pairs. They're codominant. So remembering codominant, you get them. They're expressed equally between mom and dad, okay? And they have a high degree of polymorphism. Polymorphism means there's a lot of variance in DNA between individuals, okay? And this is because my short tandem repeats means that these 2 to 6 base pairs are repeated multiple times on a chain. Okay, and the number of times it repeats varies largely throughout the population. So there's a high degree of polymorphism. So, and we use this a lot in forensic cases and in paternity testing, like on Maury. You are not the father. Okay, so they're using short tandem repeats to check that. Okay, so what does this all mean? Okay, so let's go back to our inheritance example. So we have a mother and a father. Okay, we're showing here STR sequences, and we're going to say that this STR sequence, all right, is GATA. So the base sequence GATA. Okay, the mother repeats the GATA 2 times on one gene and 3 times on the other, and the father repeats the GATA sequence 3 times on one gene and 5 times on the other. Okay. So like I said, mother has 2 repeats of the GATA on one and 3 on the other, while the father has 3 on one and 5 on the other. Okay? All right? Even if they were the same color, there would still be a visible difference between them, okay? This is measurable. It's the fact that this is measurable that makes STRs so valuable in forensic science. Okay, so your inheritance. Now, we see if we have two people who have these children, all right, we know where the STRs are in the ranges in the non-coding DNA. So we know where to locate STRs, all right? And that we also know that each person gets one half from their mom and the other half from their father, okay? And so this table, the Punnett square, shows us all the different possible combinations that we could have of STR lengths for offsprings from this mother and father. Okay, and it's this very simple inheritance that makes it useful in studies of genetics and paternity. Okay, so here we're looking at a gene VR, okay, and then we look at the lengths. Here we have the ladder. The ladder shows us the particular lengths, okay, so the mother so let's look at the child. They have a 10 repeat sequence and a 3 repeat sequence. The mother has a 20 repeat sequence and a 10 repeat sequence. So we know that the son got his 10 repeat sequence from his mother, okay? So his other gene has to come from his father, okay? So could it come from not father? Well, obviously his name is not father. He does not express a 3. Okay, so a 3 repeat sequence, so he's not the father, okay? Did it come from this guy? No, he doesn't express a VR3 repeat sequence. But this guy right here, he could be the father, okay? All right, so that's the way they would look at this to determine the paternity. All right, there's a large number of STRs on the Y chromosome, all right? And this allows us even more to test sons and fathers using the Y chromosome for STRs. All right. So any STRs on this, they must be male. So we can test only males for Y chromosome STRs. But if you look at the number down here, this is the line is the Y chromosome, and these are all the STRs. Okay, that is a ton of STRs on the Y chromosome. All right, the Y chromosome, because it just comes from the male, they're highly conserved from generation to generation. Okay, and a man will only inherit his Y STRs from his father, just like all children only inherit their mitochondrial DNA from their mothers. So confirmation of a son's father is very easy using Y chromosome STRs. But also they can use to point to suspects, but it usually will not point to a single suspect, but it can narrow it down to one family, okay, one family who all have similar Y chromosomes, because you're just looking at the males within that family, and they will all have the same Y chromosome. And so here's a Y chromosome pedigree of a family. So this father gave his Y chromosome to his son, who gave it to his sons, and this one gave it to his son, and this one gave it to his sons. Okay, so we can determine that this is the family that the murderer or the suspect must have come from, because the Y chromosome DNA will be almost exact. The Y chromosome STRs will be almost exact for this whole family.